Hello, everyone. My name is Marjorie Roskes. I'm from the Corona Lab at Weill Cornell Medicine, and I'm very grateful for the opportunity to talk about my work on the evolution of epigenomic and genomic heterogeneity in prostate cancer from tissue and liquid biopsy. Prostate cancer is the second most common cancer in men, and it's largely driven by the overexpression of the androgen receptor, or AR. And for this reason, the first line of therapy is generally chemical or surgical castration, after which many patients do well for a period of time. Unfortunately, most patients relapse into a late-stage state of disease called castration-resistant prostate cancer, or CRPC. At this point, patients are treated with next-generation AR signaling inhibitors, after which they do well for some time, but eventually acquire resistance to these therapies as well. And this remains a major clinical challenge. CRPC is a very aggressive late-stage form of disease that's highly plastic. And histologically, it has two main subtypes. That's adenocarcinoma and neuroendocrine prostate cancer. But previous work in the lab that was published in Science in 2022 used ATAC-seq and RNA-seq from cell lines, from CRPC cell lines, organoids, and PDXs to show that there are actually 4 epigenomic subtypes of CRPC. On the right-hand side is the clustering of the cell lines and organoids, and you see the 4 clusters. This is based on chromatin accessibility. And the same 4 clusters are also recapitulated by their transcriptomic profiles from RNA-seq. And so the four groups are the following. We have CRPC-NE, which has neuroendocrine-like features and is associated with the neuroendocrine histology. There's CRPC-AR, which is driven by the AR pathway and is associated with the adenocarcinoma histology. And then there are these 2 other subtypes, WNT and SCL, which are associated with the adenocarcinoma histology, but are in fact AR negative. So CRPC-WNT is driven by the WNT pathway, and CRPC-SCL has stem cell-like features. The clinical challenge is that in order to identify these subtypes in patients to get a more granular subtype identification is that these can currently only be detected based on tissue or ATAC-seq, RNA-seq or ATAC-seq, which comes from tissue. And that is very challenging to acquire all the time because it's, number 1, invasive, and number 2, not always clinically indicated. And so this remains the identification of these subtypes remains a clinical challenge. And so for this reason, we turn to cell-free DNA to try to address this issue. So when a cell undergoes apoptosis or necrosis, DNA is released from the nucleus and enzymatically digested as a cleanup mechanism. However, nucleosome-protected regions of the genome escape this degradation mechanism and escape to the plasma as free-floating fragments of DNA, which we call cell-free DNA or cfDNA, such that if you would do a whole genome sequencing of cell-free DNA, shown this schematic on the right-hand side, what we see is peaks in regions of nucleosomes and valleys in regions of open chromatin. And the dominant fragment length in cell-free DNA is 167 bases, which is the length of the DNA wound around the histone bundles. Moreover, the peaks are spaced at 190 bases apart, which is the distance between nucleosomes in closed chromatin. And so you see this very strong enrichment of nucleosomes in cell-free DNA from whole genome sequencing. And this bottom plot schematic here is just to show that if you would look at the same region in whole genome sequencing of tissue, you would see no such peak and valley signal. So why are we interested in cell-free DNA as a mechanism to address this issue? The first is that the collection is fast, inexpensive, and non-invasive, which allows for serial collection. It also captures the information from the different metastatic sites. And also, when we do whole genome sequencing, it not only gives the genomic information, but because of the special feature of cell-free DNA that it's enriched for nucleosomes, we can infer the epigenomic information. So with that in mind, we have a cohort of several patients for whom we have whole genome sequencing cell-free DNA and or matched tissue RNA-seq. So we have 25 of our own in-house cell-free DNA samples that we sequenced ourselves, and we also supplement with publicly available data for a total of 44 cell-free DNA samples from adenocarcinoma and 16 from neuroendocrine. And we also have 313 tissue RNA-seq samples from adenocarcinoma and 16 from neuroendocrine patients. And just to reiterate, for a subset of these samples, we have matched whole genome sequencing of the cell-free DNA and tissue RNA-seq from and tissue RNA-seq, and that's time matched. So in order to identify the fractional contributions of each CRPC subtype to the patient's tumor profile, I decided to shake things up a bit, and I'll talk about deconvolution. So we do deconvolution on the tissue RNA-seq first. The first step is to build our signature matrix. So to do this, we use in-house CRPC organoids and cell lines, where the goal is to generate a signature matrix where we have CRPC subtypes in the columns and genes in the rows, and each individual index represents the signature expression of that gene in that particular CRPC subtype. We then use that to estimate the fractional contributions by taking a mixture matrix where we have the gene expression for our samples. This is from tissue RNA-seq. And we don't know what the fractional contributions of these samples are. We deconvolute it back onto the signature matrix and we get out those fractions. And so we did this for 2 tissue RNA-seq cohorts. That is the Stand Up to Cancer cohort and an in-house Weill Cornell cohort. And the way we read these plots is that each individual row is a patient, and the length of a particular color represents the fractional contribution of that particular subtype to that patient's tumor profile. And we see that CRPC-AR and CRPC-NE tend to be more homogeneous than CRPC-SCL. That is to say that CRPC-SCL often appears in the presence of other CRPC subtypes, which points to the transitory state of this subtype. Moreover, the dominant subtype distribution is consistent across cohorts. So if we assign the dominant, what we wanted to see if there were particular sites of metastasis that were enriched for particular CRPC subtypes. So what we did was we assigned the dominant CRPC subtype to the patient, and we found that CRPC-AR is enriched for lymph node, CRPC-NE is enriched for liver and soft tissue, which is consistent with previous findings. And we also found that CRPC-SCL is enriched for bone. So next we want to do the same thing by estimating the fractional contributions of each CRPC subtype, but in the cell-free DNA. So the math is very similar, but the intuition is a little bit different. Instead of using gene expression, what we want to do is use chromatin accessibility that we get from the cell-free DNA. And so we use CRPC PDXs and healthy controls to build our signature matrix. So in this case, we use our healthy controls as an anchor to anchor our feature space on that, so that we make sure that we're only looking at the CRPC specific features. And then what we do is we end up again with our signature matrix, which has subtypes in the columns and features. In this case, it's not gene expression. We look at the coverage in subtype specific chromatin accessibility sites. And so we have coverage in those regions. And we do the deconvolution by having our mixture matrix. So again, these are our whole genome sequencing from patient samples. We don't know their fractional contributions. We just know their coverage in subtype specific chromatin accessibility sites. And we deconvolute that back onto the signature matrix to get fractional contributions. And here I'm showing a comparison between patients for whom we have matched tissue RNA-seq and whole genome sequencing cell-free DNA. So left-hand side is the RNA-seq, right-hand side is a cell-free DNA. And we find that the dominant subtype is always captured by the cell-free DNA, although sometimes smaller contributions can be missed in the cell-free DNA, and we're currently estimating our limit of detection. We always find the dominant subtype. And we also found that the fractional components are in fact quite similar between tissue and cell-free DNA, which suggests that the dominant subtype across the metastatic sites is the same. So here what I show is the fractional components of each CRPC subtype in cell-free DNA, but these are patients for whom we have only whole genome sequencing of the cell-free DNA. And I stratify them by patients for whom we have 1, 2, and 3 time points. And we again have the very similar dominant subtype distributions, and we are also able to track the fractional contributions over time. So that's the use of having cell-free DNA, that we can have serial collections. You can see in many of these patients, the dominant subtype does in fact change from one time point to another. For example, I'd like to point out this patient over here, AE180, who at baseline is 100% CRPC AR. But at progression, we see that he is developing into a neuroendocrine-like prostate cancer. He has about 50-50 AR and NE, maybe 60-40. By the time he arrives at progression 2, he's 100% NE. And so we're able to track those the changes in the patient's tumor profile by using this fractional contribution estimation. Also, I put here the histology of the patient next to each individual patient, so that which is binary neuroendocrine or adenocarcinoma, to show that these patients are being treated as many of these patients, especially the ones with adenocarcinoma, are assumed to just be CRPC AR, and they're being treated with RCs. But here we show that the adenocarcinoma histology is actually quite heterogeneous on the epigenomic landscape. And previous work in the lab has, I show here the Kaplan-Meier curve on the right. The previous work in the lab showed that patients with SCL do not respond to RCs as well as patients with CRPC AR. And so this can hopefully be used in the clinic to guide patients' personal therapeutic strategies. And so now that we have the epigenomic fractional contributions for each patient at a given time point, what we want to do is leverage the fact that we have whole genome sequencing cell-free DNA. So we still have, this is based on the epigenomics, so the chromatin accessibility, but we still have we did whole genome sequencing, so we still have the genomic information. So what we want to do is look at genomic events to see if we can see consistency between genomic events that are happening that are genomic events that are happening, and see if we can see consistency based on the fractional contributions. So with that, I want to show some examples of congruence between the genomic events and the epigenomic subtypes. So here on the left-hand side, I'm showing this patient, DTB102, who is histologically an adenocarcinoma. Epigenomically, he is 100% CRPC AR. And genomically, we find that this patient has amplifications at the AR gene and the AR enhancer, and he also has a missense mutation at the AR gene. On the other hand, we have this patient, DTB097, who has an adenocarcinoma histologically. However, he has 0% CRPC AR epigenomically, and he is quite heterogeneous with the other samples, where the dominant subtype is CRPC SCL. Genomically, he has no amplifications of the AR gene, and he has a heterozygous deletion of RB1, which is associated with the neuroendocrine-like subtype. And then we also, interestingly, we found that he had 16 out of the 19 hippo core genes are amplified. And this is significant because The Hippo pathway is very important in SCL because it influences the stem cell behavior, and dysregulation of this pathway can enhance stemness. Also, the Hippo pathway regulates YAP-TAZ, which has been shown to be very important in mediating the transition to the SCL subtype. So here are some more examples for patients for whom we have more than one time point. So on the top row, we have AE015. At baseline, this patient is very, very heterogeneous. He is a little bit SCL, a little bit AR, a little bit NE. We find that he has no amplification at the AR gene or the enhancer, but he does have deletion of RB1. At progression, however, CRPC AR is dominating epigenomically. He's now almost 70% CRPC AR. And now we see amplification at the AR gene and the AR enhancer. And we still see deletion of RB1, which is associated with the neuroendocrine subtype. On the bottom row, we have this patient AE124, where at baseline, he's 100% AR. Genomically, we found 4 missense mutations at the AR, and by the time he's at the progression time point, he becomes very heterogeneous. AR is no longer as dominant, and the existence of other subtypes appears, and we no longer see any mutations in the AR, suggesting that a different clone has emerged. And so with that, we're able to show that there is consistency between the genomic events that are happening and the epigenomic events that we're able to identify the fractional contributions and really leverage all there is to offer from whole genome sequencing of cell-free DNA, hopefully to influence the treatment protocols that clinicians can have in these patients that may actually be quite heterogeneous. So with that, I'd like to thank everybody who contributed and guided me in this work. And I would especially like to thank the patients and their families who contributed their biological information to this project, and we wish them a speedy recovery. I'd also like to give a shout out to my colleague Wei Ling, who had a poster yesterday, A133, on predicting tumor gene expression from cell-free DNA whole genome. If you didn't get a chance to see it, make sure to check it out online. Also, the lab is hiring, and if you'd like to join, please feel free to reach out. I'm happy to take any questions, and thank you for listening.
All right, we have a couple minutes for questions, so I have a question. I will go you. All of this is for castrate resistance. Is there any evidence that in prior stages of the development of prostate cancer, there's also epigenetic differences? Because once the patients are treated with castrate, surgery, they are kind of in the path to evolution to this. It would be perhaps interesting to see if there's other treatments that can be applied earlier in the stage.
Mm-hmm. So I think that primary prostate cancers are generally thought to be the AR, which is why our first line of treatment is like androgen deprivation therapy. But in this particular study, we only dealt with patients with metastatic who are already in the metastatic state. So, but yeah, it would be really interesting to look at patients who are in primary states.
All right, another question here.
Actually, I think it would be challenging to look at patients from cell-free DNA to look at patients who are in primary disease because they don't have as much tumor DNA shedding to the cell-free DNA. So it would be, I think, quite challenging to be able to answer that question using cell-free DNA.
Hi, that was a really nice talk and very, very nice work. I had a practical question. What sort of sequencing coverage do you need on the cell-free DNA to be able to do your analysis?
Yes, so I didn't have time to talk to add that in the slides, but we did do power calculations to show that we can go down to about 4 to 5x coverage in order to confidently estimate the same fractional contributions. These samples were sequenced at between 40 and 180x, but we did the power calculations to show that you can go down to about 4x.
Another question?
Yeah, thank you so much. That was a really interesting talk. First question was just, what kind of lower limit do you see for this modeling with respect to tumor fraction and the cell-free DNA?
Yes, so we can go down, we did the power calculations for the tumor fraction as well, and we can go down to about 0.3.
Okay, about 30% tumor.
Correct.
Okay, so still fairly high. And the as for the actual modeling itself, when you're inferring, trying to go back from inferring the protection score basically for those fragments that are enclosed by nucleosomes, is that like a windowed protection score? Or are you doing some other kind of inference to get your signatures matrix?
So we actually use the coverage directly. So there are, I mean, you can use the window protection score to identify nucleosome peaks, and that's from Snyder et al. From Cell 2016. But we use the coverage directly so the previous work in the lab that used HiTSeq in cell lines and organoids, they had reported subtype specific chromatin accessibility sites. So we look at the coverage in cell-free DNA at those subtype specific sites, with the understanding that if the region is open, then there should be lower coverage in cell-free DNA. And if the region is closed, there should be higher coverage in cell-free DNA because of the nucleosome enrichment.
Yeah, okay, that makes sense. Thanks very much.
Sure.
And there's a last question there online. Go ahead.
Yes, the last question. We have a question online. First of all, Marjorie, thank you very much. It was a very nice talk. The question is asked by Zheng Deng Lei. Sorry if I mispronounce it. But anyway, when comparing baseline versus progression, the difference is due to the intratumor heterogeneity, or is it due to a progression of the disease?
I'm sorry, can you repeat the question?
Okay. When comparing
Yeah.
baseline versus progression, the difference is due to the intratumor heterogeneity, or is it due to the progression of the disease?
I think can be both. I think can be both because the different metastatic sites contribute, but likely progression as contributes a big portion of that as well.
But thank you again to the